June. And we've sort of made it up through gastrulation to the beginning of neurulation and beginning to think about how embryos acquire patterns for their body in terms of establishing body axes, anterior, posterior, dorsal, ventral, and left, right. And the way I have the course arranged, I'm going to pause on the vertebrates and now take a sideways step over to think about a couple of other genetic systems, model systems that we use to study these kinds of events in developmental biology. And this will give us a chance to kind of compare and contrast how different organisms meet this challenge of setting up a body axis. And you'll see that there are some similarities in the way that they do it, but also some distinctive, unique properties as well. Also, it gives me a chance just to introduce to you a couple of different organisms that scientists use to study these things and to sort of highlight what are the serendipitous features about these creatures that make them really useful tools for developmental biology. So today, we're actually going to talk about two. The first one is the nematode worm, called C. elegans, and then the second one will be Drosophila, which is a very, both of them are really neat organisms. So keep in mind this general principle here, sort of underlying all the themes and the significance of what we talked about, where we have a gradual restriction of cell fates as time goes by within the embryo. So with every cell division, whether you can see it morphologically or not, the embryo is gradually paring down for one daughter cell at a time. What is the range of possible fates that this could acquire? And of course, you could argue that the really interesting bits are happening here at this stage where the cells actually do make some kind of decision. They make a commitment to one cell fate or a choice of one cell fate versus a different one. So this is the nematode worm C. elegans, which full Latin name is Cyanorhabditis elegans. And I think you can see why people like to abbreviate it, C. elegans. This is a small worm, about 1.2 millimeters for the adult length. It lives out in the soil. It eats bacteria. It has really not much economic importance to man at all. But for developmental biologists, it's turned out to be a really useful model system for looking very, very early as cells divide and learning something about how they're separating out the potential for different daughter cell fates. One of the things you can see is that these eggs and the adults even are transparent. So that's kind of handy for being able to observe some of the cells as they develop inside the embryo and maybe move to new locations or take on different shapes. Development is pretty rapid. So from a fertilized egg to an adult in only 3 days. So you could conceivably do a multi-generational genetic scheme if you were trying to breed a certain mutant phenotype, for example. The lifespan is short, only 2 to 3 weeks. They're really easy and cheap to grow in the lab. Basically, all you need are some little agarose petri dishes with some bacteria on them. You can freeze them down, you can thaw them out, so it's real easy to keep these. And compared to mice that cost hundreds of dollars a day to maintain the colony, these guys probably a couple $100 for the whole year could provide for your animals for the lab. Okay, so from fertilized egg, they're going to go through 4 different larval stages where they grow and molt each time, and then they'll become an adult. And here on this panel, you can see from the 1-celled embryo on top, 4 cells, and then as this thing starts to divide, we get to the comma stage, the pretzel stage, and then finally they'll hatch out of their eggshell and be a true worm crawling around the dish. So I think I will pause here and show you a video of the C. elegans development. Once again, it's not showing up on my screen. Tell me. So right now you can see the one-celled embryo, and this little border here, this is the eggshell. So within the confines of a fairly stiff eggshell, these cells will begin dividing. So there's our first two pronuclei, a male pronucleus and a female pronucleus. So immediately after fertilization, the sperm gives a nucleus and the egg already has a nucleus. And these two have to find each other, meet and fuse to recreate the diploid single nucleus of the zygote. Okay. All right, so there they are fusing. Now we're going to see our first cell division. That's the mitotic spindle that got set up there. You can see the two poles of it. And here's our first cleavage furrow within this embryo. Okay, one daughter cell divided, and there goes the second one. There are 4 cells. And you see they didn't really grow that much. They're just dividing up this large starter egg into smaller and smaller cells as time goes by. And that shows you the first several divisions for this. Okay. All right. Okay, so here's what the adults look like. And C. elegans comes in 2 sexes. One is a hermaphrodite. And as you might expect, this hermaphrodite can make both sperm and eggs. And yes, the sperm from the same individual can fertilize its own eggs and make what's called self-progeny. So these progeny are coming entirely from one individual. We also have a male sex, and males can mate with the hermaphrodites and give progeny that incorporates 2 different genomes, the male's genome and the hermaphrodite's genome. So here you can see some of their internal anatomy. On the outside they have kind of a cuticle structure and some muscles within this body wall that enable them to crawl around the dish. In the interior there in yellow you see the gut tube. So this is going to involve a mouth, a pharynx is a region where they grind up the bacteria, a long gut tube for absorbing these nutrients, and ending in the anus on the other side. And then most of the rest of the worm is actually dedicated just to reproduction. So they have a huge gonad inside of there, and they're able to make both eggs and sperm, fertilize them internally, and then you're going to get the first several divisions for those new fertilized zygotes within the mother, within the body of the mother. At some point then, these new embryos will be laid or deposited outside of the mother through the vulva opening. Okay. So these hermaphrodite adults, it was found, have exactly 959 somatic cells. So 959 cells for the body, not counting how many oocytes or sperm that individual may produce. Just a little bit about the males. They're really similar to the hermaphrodites, but they're slightly smaller and thinner. And on one end, they'll have this tail apparatus, which is sort of shaped like a hook. And when 2 worms are mating, what will happen is the male slides up alongside the hermaphrodite and hooks that male tail into the vulva opening. And then here in the anatomy of the tail are little ducts that the male can transfer sperm through these ducts into the hermaphrodite. And then those sperm actually move and become stored inside of the female or the hermaphrodite's body and can be used to fertilize eggs as they come down the pipeline, as it were. So Dr. Sidney Brenner, he is one of the founders of using C. elegans for developmental and genetic studies. And so I just threw his name up there just to kind of give you a flavor of who some of the people are in this field. So the gonad, or the reproductive structures within this hermaphrodite, are shaped like two lobes. So here is, in this box, one of the lobes shaped kind of like a U on one side, and then you can see a second one up there on the other side folding around. At the very tip of this gonad is the distal tip cell, and this is a stem cell that gives rise to many mitotic divisions that are going to give rise to all the oocytes as they form. And so initially, you see all these nuclei, and then they'll eventually form into haploid cells. And they're sort of lined up here in a pipeline heading down towards the vulva. And they're going to pass through this little chamber here, which is known as the spermatheca. And that's where the sperm are. And as they pass through there, they'll be fertilized. And then you can see here, these embryos are starting to divide and go through the first couple rounds of cleavage while still inside of the mother. Then at that point, they'll be laid in the dish or the environment external to the mother. Now, this is a picture of sperm. So these sperm are present there in this little spermatheca chamber, and the eggs are kind of coming to them. They're just sort of going down the pipeline and passing right through the chamber. And interestingly enough, the sperm are very different in morphology. They don't have a long tail like we typically think of with sperm, but on the other hand, they don't really need to swim anywhere. They're already right there. So they can probably just crawl on over and fertilize the next egg that came down the pipe. Okay, another key player, Dr. John Sulston. Sometimes people do these experiments or projects that are really simple but become incredibly foundational for the field. And this is the case for his work. What he did is he sat down with a microscope and some fertilized C. elegans and just watched how they divide from one individual to the next. And as a matter of fact, he actually tracked from the 1 cell stage to the 959 cell stage where and when each cell divided and in what orientation. And he made this map to reflect that process. So up there on the top you see the zygote, and then you have a lineage tree of how that zygote divided into 2 cells and how each daughter cell divided as time went by. And then on the vertical axis you have time. So you'll see that within the first few hours there's a huge number of divisions, and then they're mainly done for most of the cells, with a few exceptions that take a little bit longer to generate their final numbers of daughter cell. Another key discovery that he made in the course of doing this mapping of cells is that if you take one individual to the next in C. elegans, this pattern and timing of cell division is exactly the same in every single individual. Now, if you think about it, that's actually going to be quite handy if you're a scientist trying to think about when and how cell fates are established. So here we have a model that is reproducible and the same in every individual, and so you could start to make predictions and start to test or interfere with different cell fates as they arise, maybe through mutants or other methods, and then learn something about how this is happening in the wild type situation. Okay. So this is known as the invariant cell lineage because it reflects this very stereotypical, very standardized, very non-varying type of way of dividing up cytoplasm and establishing a lineage that gives rise to all the different daughter cells of the adult animal. So if we just take a somewhat more simplified version of this, we can think about what are the cells that might be considered founders for different lineage types, such as, is there a cell that is the ultimate grandfather cell that will give rise to all the gut? Is there a cell that will give rise to all of the muscle? Is there a cell that might give rise to all of germline? And in some cases, yes. So here is a cell which happens to be named E, and this does give rise to the entire group of cells that form the intestine, which there's also the endodermal layer. But when you look at other cases like neurons or muscle, there might be more than one founder cell that gives rise to that lineage. In the case of the germline, there is an actual cell called P4, it's right here, and it will give rise to all the germline, including both sperm and eggs. These founder cells, the idea that they could exist and that we could find them, that's also interesting because what is it that these cells are doing in terms of differential gene expression that might make them into the founder cell of a whole lineage? For example, is there a factor that that you need to make endoderm or gut cell fate in this organism, and what is that factor? Maybe that factor would be the same in the fruit fly or the frog or the human. So it would just be interesting to know what these genes are. We have a system here that we could do genetics with. We can find mutants, and we can then begin to figure out through the process of genetic what these genes are doing and which genes are the important ones. So in terms of cell fates, this does get divided fairly rapidly within the embryo. So here you can see already at the 4-cell stage, some decisions have been made. Cell fates have been parsed out differently among the 4 different daughters. And then ultimately down the line, each of them is responsible for building a different part of the embryo. And so this means that we have here an organism that we could look really early and think about what are these really early events that might make it possible to divide the future fates in this fashion. How can this be done spatially? How can it be done so quickly? And how can it be done so reproducibly? Another kind of interesting thing that really caught my attention when I first heard about these worms is that one of the cell fates is actually to die, for the cell to die. So this is called programmed cell death. And if you actually look from the original zygote cell to all the daughter cells that are generated, you had actually generated 1,090 cells, but 161 of them, it's their cell fate is to die. So these cells die at a very particular time, in a very particular place, and it's always the same exact cells that die in each individual. So if any of you are taking genetics this summer, maybe you're starting to think, maybe there's a gene for that. It's sort of like there's an app for that. Well, there's a gene for that. And this is Bob Horwitz, and he has done these studies using genetic screens to find phenotypes where now you get too many cells are dying, so that's not normal, or too few cells are dying, which is also not normal. So what is it that can cause exactly 161 cells to die? Well, he found an entire pathway of genes that cause this gene-regulated cell death or programmed cell death in the embryo. Now, at the time that he found these, we thought, well, this is kind of like a special feature for C. elegans, and maybe it will teach us about some mechanisms in this organism. But humans don't really do this in the same way. We have a totally different mode of generating cells and killing cells. But it does turn out that some of the same genes in this pathway are the same genes that in humans or other organisms are part of the death-killing mechanism or part of the programmed cell death mechanism for these cells, for these organisms as well. Programmed cell death and apoptosis, you might have heard that term, are now considered to be basically the same thing. They're both directed type of death based on gene expression, not injury or anything like that. In the embryo, you can recognize these cells that are about to die because they sort of swell up and become round and prominent, and then they'll disappear shortly after that. Okay, so one of the things with C. elegans early cleavages is you can see that these events are very, very programmatic. That is, they're very set in stone and exactly the same from one individual to the next. So when we see the first cleavage forming in the fertilized embryo, it always forms in this fashion to generate an AB daughter cell that's slightly bigger than the P1 daughter cell. Is that important? Well, it turns out you can find mutants that generate 2 daughter cells exactly the same size, lethal. So already exactly where you put that cleavage plane is going to be totally very important for survival for this embryo. Next, we get the second cleavage, and you can see the AB cell is dividing a little bit earlier than the P1 cell. But the P1 then catches up right after that by undergoing another division. And these always happen in the same orientation and with the same timing. So timing is also an incredibly important factor when you think about development. Exactly when something happens can often be as important as where it happens in the embryo, and can also be something that's controlled by genes. So we're going to end up here with these 4 cells in this orientation. And if you think back to the original one-celled organism, and imagine that there might be some maternal factors in this thing that are not evenly spread out throughout the cytoplasm, but might be kind of more in one side or the other side. If we always divide that initial organism in the same fashion, then reliably each daughter cell would inherit that portion of the cytoplasm that might contain those maternal factors. This will happen every single time. So in C. elegans, if we think about when does the zygotic transcription initiate, that is, when do you start to get mRNA made from the genome of the zygote and its daughter cells, it does not happen until we get to about the 8 to 12 cell stage. So this is one of the characteristics of the mid-blastula transition, that as organisms pass this kind of marker point in time, now they're going to be much more robustly expressing genes from their own genome as opposed to just relying on factors from the mother. So if this is not happening until 8 to 12 cells in the worm, it must mean that it's mainly maternal products that are controlling these early events. So maternal products, mRNAs, and proteins put into the egg by the mother, those would be the genetic factors responsible for all of these events and for, making sure that the organism is doing the timing and the orientation of the cleavages in precisely this way. So indeed, C. elegans does rely heavily on these maternal factors to control these early events. So you can also do genetic screens to try to find what are these factors or what are these genes. And this is some work done by Ken Kemp, who's at Cornell University. And actually, I have done my PhD at Cornell University, and as part of our program, you have to do 3 rotations where you spend some time in different labs. And so I got to do one of my rotations with Dr. Kemp and work on C. elegans. And he had already done some of this work, but I had a little follow-up project on one of the genes, so I've always felt like this was really interesting to me, and I sort of wanted to see where it would go over the years. So his main principle here is to ask, what are the genes that are needed to control how these cleavages happen within space and time, and what are the protein products that those encode, and what do those proteins actually do? Now, it's a little bit tricky because you're thinking actually about getting a mutation in a gene that's expressed in a mother making eggs, right? Because the mother has to make these factors and put them in eggs. So you're really looking for a very special class of proteins or genes that act at that time in development. But in principle, you can apply any kind of x-rays or chemicals to cause mutations randomly across the genome. Then you're going to do some screening. You're going to look through eggs from various different mothers and see which ones have interesting phenotypes. So you need to define what interesting might look like. And then ultimately, you can use genetics to track back and map that gene and figure out what actual DNA sequence was altered here, and what does that gene encode, and what does that gene do functionally for the embryo. So he did this, and he found a series of genes that we call the PAR mutants. And I'm going to tell you about those here in a second. But I want to introduce one other thing here, and this is these moieties. These are actually protein RNA complexes found in the early embryos that are called P granules. And this is another one of these little serendipity stories in science. So somebody was trying to make an antibody to recognize a protein that's found early in C. elegans. And so, you know that to do immunohistochemistry, you have to have a unique primary antibody for your protein of interest. And this is what they were trying to generate. Well, it didn't work. They got back an antibody that recognized something else. But this something else turned out to be really interesting. And these are the P granules. And so what you see here is an embryo that's at the 1-cell stage, 2-cell, 8-cell or so, and then multi-cell in these different panels. And in the 1-celled organism, this antibody is recognizing something, some kind of factor, and you can see it's almost all over to one side of that initial fertilized egg. And then as cell division goes through, that factor labeled by this antibody gets segregated into one daughter cell only, which happens to be the P cell. So even at this stage down here where we have maybe 50 different cells, that one daughter cell has inherited all of the P granules that are being recognized by this antibody. So the P lineage is what gives rise to the germline. So here we have, sort of by accident, we found a reagent that can recognize a special maternal factor that gets segregated out during cleavage, sequestered into one cell, and that is the cell that becomes the germ. So, pretty interesting. So these P granules, we can call them a morphogenetic determinant. And Susan Strom is the lab that found that antibody. And here you can see the lineage for early C. elegans, that is the pattern of cell division giving rise to these P4 germline cells. And every time one of the daughter cells does not inherit those P granules, and they give rise to a variety of different cell fates, and then finally P4 becomes the germline. So this concept here, this term of a morphogenetic determinant, sometimes called a cytoplasmic determinant, really it means the same thing. And this just refers to some kind of a factor, some kind of a gene product, protein or RNA, and the presence of this factor in the cell actually determines what fate it would acquire. So far we have really a show it or a correlation here where we have the presence of this factor and the development of the germ cell fate. We don't know if the factor causes the germ cell fate, but that's a nice hypothesis to test. So one way, just think more generally, how do we assign cell fates? One possible way of maybe doing this is having factors that would be key players for key cell fates, and if the cell gets that factor, well then that is the start of their process of differentiation towards that particular cell fate. That could be a way of doing things, and in general we have found a lot of scientific evidence to support that sort of model, at least for some cell fates in some organisms. Okay, here's the genes from the Ken Ketley's screen, which is also done by Bill Woods, who's a researcher over in CU Boulder, who's been working on C. elegans for a long time. So these PAR genes, PAR stands for partitioning defective because they partition their cytoplasm in an inappropriate fashion. And the function of these genes, based on mutant phenotypes, is to coordinate cell division and the localization of factors within the cytoplasm like these P granules. And there are several different genes with pretty similar but slightly different phenotypes within the C. elegans embryo, but they all have some sort of problem in these three areas. The first is how they divide the cytoplasm in division number 1. And typically the PAR genes are going to make the daughter cells, 2 daughter cells that are exactly the same size, okay, as opposed to the slightly larger AB cell in the wild type. Then I was telling you about how the orientation and the timing of the spindles for the first several divisions is very stereotyped or very identical for each individual. In these par mutants, that's changed in some sort of subtle way. So for example, these two, the two cells here are dividing to get to the 4 cell, they're dividing exactly the same time. And that's a subtle difference from wild type, but turns out to be important. And then another characteristic feature is if you look at how these P granules are segregated, so at how we're actually dividing up the cytoplasm, that is not the same as in wild type. And here you have multiple different daughter cells are inheriting some of the P granule cytoplasm, as opposed to just that one P lineage cells. So the P granules are being missegregated. And probably not just the P granules, right? Anything else that's in the cytoplasm is not being divided to the daughter cells in exactly the same way as you find in wild type. Okay, so all of the par mutants are lethal. So messing this stuff up early on causes havoc in the embryo, and you are not going to be able to get the right number of cells, the right number of cell fates, the right timings or positioning physically. All that stuff from this point on is going to be different than wild type, and eventually that accumulates in the death of the embryo. Okay, so I've kind of been talking about genetic screens, and I just wanted to take a slide here to make explicit exactly the logic of how this is kind of working. And I think that you guys already know this, but I think at the same time it's also can be helpful to just lay it out there as exactly what's going on here. So when you start out with a process of interest, like how cell fates are being established, genes can help find a handle to discover the mechanisms or the workings behind this. So you can start out by looking for phenotypes of interest. So we're interested in how these cell divisions are happening, how you might segregate P granules or morphogenetic determinants. So let's look for mutants that don't do that correctly. They divide differently. They segregate the P granules.